Hello, my name is Alexandra Goretzki from the Paul-Ehrlich-Institut, and I would like to shortly introduce our paper entitled Stimulation of naive B cells with a fusion protein consisting of FlaA and Betv1 induces regulatory B cells ex vivo. In type 1 allergy development, the uptake of an allergen by antigen-presenting cells triggers the differentiation of allergen-specific naive T cells into TH2 cells via the cytokines IL-4 and IL-13. These cells, in turn, via the same cytokines, promote the activation of allergen-specific B cells that undergo an isotype change, resulting in IgE-producing plasma cells. The secreted IgE binds to the high-affinity Fc-epsilon receptor on mast cells. Upon second contact with the allergen, cross-linking of IgE on the surface of the mast cells result in the mediator release, causing the typical allergic symptoms and the potentiation of the reaction in a positive feedback loop. In this study, we have looked at the effects of a potential allergen therapeutic, rFlaA Betv1, consisting of the TLR5 ligand, the bacterial flagellin, and the major birch pollen allergen Betv1, that we developed to improve the treatment of birch pollen allergy. Our work focused on the effects of rFlaA Betv1 on ex vivo isolated B cells. We could show that this fusion protein strongly and T-cell independently activated ex vivo isolated B cells, inducing the formation of a B-cell subset, characterized by the expression of the regulatory markers CD1D, CD24, as well as IgM and CD38. Additionally, these regulatory B cells also secreted the anti-inflammatory cytokine IL-10, which was found to be TLR5 independent but MyD88 dependent. In a co-culture of these fusion protein-stimulated B cells with dendritic cells and allergen-specific TH2-biased T cells, the B cells were able to suppress Betv1-induced TH2 responses, shown by the reduction of allergen-induced TH2 cytokines like IL-13. In the context of the allergic pathomechanism, this may interfere with the differentiation of further TH2 cells and reduce the formation of IgE-producing plasma cells. As activated B cells also produce antibodies, we finally checked for the antibody secretion upon stimulation of the cells. Here, stimulation with a fusion protein resulted in the production of IgG antibodies, which were shown to bind to the allergen Betv1. In theory, these antibodies have the potential to prevent the cross-linking of the IgE on the surface of the mast cells and thereby either reduce or prevent allergic reactions. Thus, in this study, we demonstrated for the first time that a potential allergen therapeutic has the possibility to induce regulatory B cells ex vivo. With these results, we hope to contribute to a better understanding how such modular vaccines and therapeutics can improve allergy treatment in the future. With this, I would like to thank you for your attention.